I would like to introduce you to my colleague, Dr. Virginia Lee, who is the Manager of Supportive and Palliative Care Program and Services and the Director at Cedars Can Support, the McGill University Health Center. Thank you.
Thank you, Rolla. So I am so pleased to welcome everybody to the Cedars Can Support educational clip. And today's topic is about clinical trial basics with our guest speaker, Dr. Stan Shapiro. It's a very, very important topic in today's educational clip for many reasons, because you'll take away knowledge that's important for yourself, but I think it can also be applied to a broader context to understand news stories that you might hear about in the public, particularly maybe also to understand the COVID vaccine and the rigor with which it went through before it went to the public. But the educational clip will also empower you to understand consent forms and ask better questions if you're ever asked to join a research study. And Dr. Shapiro will also let you know, give you information about, it's not just about drug trials, but it's about quality of life studies, or even if your child is asked to join a research study in school, you'll have better knowledge and you'll be better qualified to ask the more appropriate questions. So there's no better qualified person to give this information than my previous professor at McGill University. But I was also very pleasantly surprised to learn that when I came on into this role at Cedars Can Support, that he's also one of our devoted Can Support volunteers in the palliative care unit at the MUHC. So you'll see why. I'm going to read his bio sketch, and you'll see that he is we're very pleased to have him join us. So Dr. Stan Shapiro is a professor emeritus at McGill University and a fellow of the Society for Clinical Trials. He obtained his PhD from Stanford University. And in addition to McGill, he held positions at Harvard University and the University of Michigan. His long-standing research interests center on applied and theoretical aspects of clinical trials. And in addition to his involvement in numerous trials, he has also served on a variety of clinical trial, scientific advisory committees, data safety and monitoring boards, and he served in a variety of capacities on the Medical Research Council Canada and the Canadian Institutes of Health Research for the clinical trials grant committees. So without further ado, I want you to get comfortable wherever you're viewing this webinar, and write down your questions or comments that we can discuss and at the Q&A at the end of the presentation. And remember that this is a webinar that's being recorded, so the slides and the recording can be available on the Cedars Can Support website in case you are wanting to refer to it at a later date. So without further ado, I'd like to present to you Dr. Stan Shapiro.
Thank you very much, Virginia. I'm going to be using slides throughout the presentation, but don't worry about trying to capture the information on the slides, as they'll be available subsequently through the CEDAR website. Although, like many of you, I've been using Zoom throughout the last 2 years for meetings and get-togethers with friends and family. This is the first lengthy Zoom talk I will have given, so I apologize in advance for any rough edges and hope you'll bear with me. It's going to be a novel experience hearing myself talk without interruption for an extended period of time, certainly not like a family Zoom. So we're going to be dealing with basics of clinical trials. So let's take a look at what we mean by a clinical trial. And basically, a clinical trial is a research study involving human volunteers in which individuals are assigned to groups to receive one or more interventions or treatments so that researchers can evaluate the effects of the intervention on biomedical or health-related outcomes. And the treatment assignments are determined by the study protocol. We're going to elaborate on this aspect and look at different ways that that assignment is made in a second. But first, we'll just briefly look at the different kinds of trials. And Virginia has already alluded to that in part, because we can use this tool, which is a valuable tool for acquiring new information in a variety of different contexts and settings. We can be looking at prevention trials in which we're trying to determine better ways to prevent a disease in people who have never had it, or to prevent recurrence of disease. And approaches may include medicines, vaccines, or lifestyle changes. And all of us are quite well aware of the efforts that have gone into the vaccine trials over the course of the past several years in dealing with approaches to try and prevent COVID-19. We also have diagnostic trials, which will be comparing different tests or procedures for diagnosing a particular disease or condition. Treatment trials, which will really form the basis of our focus this morning, testing new treatments or new combinations of drugs, or new approaches to surgery or radiation therapy or devices. And quality of life trials or supportive care trials, where we're looking at exploring and measuring ways to improve the comfort and quality of life of individuals with certain conditions or illnesses. Okay. So generically, as a model to be keeping in mind, we can think of drug trials. That will probably be the simplest for our purposes. And in that context, there's an additional nomenclature that gets used for different phases of trials. So this graphic may look a bit complex, but it's really basically just a graphic aid to help us in distinguishing between phase 1, phase 2, phase 3, and phase 4 trials. On the horizontal axis, we have time, and on above it, we have a listing of 1, 2, 3, or 4. And on the vertical axis, or the incline, we have different types of study, moving from human pharmacology for phase 1 studies. And I want to direct your attention to the diagonal and the colored in discs. So the first study of agents in humans will involve studies designed to try and examine a route of administration and an appropriate dosage and safety for a new agent. And those are phase 1 studies. Moving on to phase 2, we're looking at initial indications for efficacy of a new agent. And if we obtain positive answers in the phase 2 study, we can move on to a full-blown clinical trial, of phase 3 comparative study, which is going to be our focus this morning. And I'll just mention phase 4 studies to you. This is really the domain of pharmacoepidemiology. Once an agent is licensed and becomes widely available, we'll have substantially more information than we would have even if we've had thousands, tens of thousands, or even hundreds of thousands of individuals previously studied in its pre-licensure phase. And that data will enable us to discern perhaps rare but nonetheless important side effects that might occur with a reduced frequency, something on the order of 1 in a million, but nonetheless be important to be aware of. We're going to be looking at controlled trials, and in a controlled trial, the assignment of treatment is carried out according to a protocol, to the research plan. Thus, we have the responsibility to ensure that these endeavors are not undertaken frivolously, that they're designed and conducted to the best of our ability, and that potential participants are provided with the appropriate information to enable them to decide whether they wish to volunteer their participation. It's important to recognize that the participants in a clinical trial are volunteers, and respecting their autonomy, individuals are free to make the decision as to whether they choose to participate or not. In order to make that decision, they have to have appropriate information presented to them so they can weigh the pros and cons of their potential participation. So informed consent is an essential element of that process. And informed consent itself is a process. It's not merely the signing of a document that is presented to a potential participant, but it's an ongoing process in which information should be provided to potential participants about the study before the decision about whether to accept the offer to take part is made. The process should facilitate comprehension of the information. Often, and it's increasingly the case, I'd say, informed consent documents, the written sort of validation of informed consent being given is based on an extensive amount of information being provided to the potential participant, which could be overwhelming. And there's an important balance between providing sufficient information, but making sure that it is comprehensible and that it doesn't defeat the purposes in the first place of providing information that can be helpful. The process should also provide the adequate opportunity for you to ask questions and to consider whether to participate. Hopefully that's built in and you won't be asked for your consent to be provided in an unreasonably short matter of time. But if you are asked and you don't feel that you have sufficiently evaluated the situation yet, don't hesitate at all to say, I really need to think more about this. And when can I get back to you with my confirmation that I will be participating, or with, in fact, the information that I'm not going to be participating. It doesn't necessarily make for the best logistical experience for the researchers, but nonetheless, they should be fully accepting of anyone's desire to further evaluate the situation before they make a decision. And in keeping with the notion that we have an informed consent process, information should be continually available to participants if in fact it would have an impact on their decision to continue participation. And this information can come from within the study itself, if certain effects become manifest that weren't known at the start, if there's side effects to the treatment that become evident as this study progresses, or it could come from outside of the study with other research that's being done that does have an impact on the merit of the present study. So individuals, even though they've provided consent initially, retain the right to withdraw from a study at any point. And wearing sort of my research hat now, I would just encourage you to realize that should you choose to withdraw from a study, and if your withdrawal is based on a decision that you no longer want to continue with the treatment to which you've been assigned, you can withdraw from treatment and yet still provide follow-up information to the investigation, to the study, which will be helpful in terms of the final analysis of the results. In Canada, a document called the Tri-Council Policy Statement, Ethical Conduct for Research Involving Humans, is what governs the conduct of research with humans. And it's the Tri-Councils are the Medical Council, the Canadian Institutes of Health Research, and then the Engineering and Research Council, and the Social Science and Humanities Research Council. And this document is one which is under constant revision. It's about to undergo its third revision, and is an ongoing living document where the expertise of a variety of individuals and the general community has been inputted to try and provide a guide for researchers in terms of how they approach their task of carrying out ethically appropriate research. Okay. We're going to be focused on the randomized control trial. So what is a randomized control trial? It's a prospective investigation, and by prospective investigation, I mean it's a study where we have treatment assigned and we go forward in time and observe the outcomes that occur after the treatment assignment of the relative effect of a therapeutic, a prophylactic, or a diagnostic intervention in humans. In a randomized trial, treatment assignment is made using a chance or probabilistic mechanism like the flip of a coin. The idea is certainly not intuitive when we consider the usual situation of an individual presenting themselves to the healthcare system, to the medical system, for help with dealing with a particular problem that they're encountering. The idea of the treatment that they're to receive being decided essentially by the flip of a coin may certainly prove to be a very atypical situation for them. So why would we consider even doing such a thing? Something so seemingly bizarre in the clinical setting as randomizing treatment. Well, properly executed randomization does a variety of things. I'm going to list three particular features, the first of which is the most important, so we'll spend a little bit of time talking about it. And that is that randomization eliminates bias from the assignment of treatment. It can be the case, it doesn't even necessarily have to be conscious, it could be unconscious, that without some mechanism removed from the investigator directly, the types of individuals who receive the different treatments under investigation are in fact different. Okay? Randomization removes that element completely by removing the investigator or the researcher from the assignment process. What is done is an assessment is made of eligibility of the potential volunteer for any of the treatments under investigation. And then what is really happening is that we're asking for consent to randomization. I don't want to get ahead of myself, but I'll just mention that one of the elements from the point of view of a potential participant in a trial is when they're asked for consent, they don't know what treatment they're going to receive. Okay? They know the potential treatments they would receive, but not the particular treatment. It would be much easier, one would think, to approach the patient for consent with the knowledge that they're going to receive a particular treatment, and then they're consenting to be in the study of that particular treatment, knowing they will be receiving it or perhaps a different treatment. However, that reintroduces the possibility of bias and will make the analysis of the results much more suspect at the end of the day. So randomization is one of the most powerful, if not the most powerful tools in our ability to eliminate bias from the assignment of treatment. It also tends to balance treatment groups with regard to both known and unknown risk factors, prognostic factors, and it provides a basis for the statistical analysis that's going to occur once we have all the data available to us. Okay, so that would be the reason for employing randomization, but there's still the question, and a very natural question of, is it right for me, and perhaps even more importantly, for my physician to leave my treatment to chance? I've sought out the assistance of this individual to help me through this problem. And now I'm being told that the treatment that I'm going to receive is not going to be the treatment that he necessarily or she necessarily feels is best, but it's going to be the treatment that's been determined by the fact that the flip of a coin has turned up heads. Well, the randomized clinical trial is really a tool that's been with us for only about 75 years. It saw its introduction in essentially mid-1900s. And initially, concerns had been raised by some medical researchers about the potential ethical conundrum that a physician would be presented with if, in fact, she had a preference as to the preferred treatment for the patient in front of her, but yet recommended or offered to the patient participation in a clinical trial. This would apparently seem like a violation of the way that the process should normally work. So let's think of an example. Mrs. Kaplan comes to Dr. Jovanovic and is trying to deal with her chemotherapy-induced nausea and vomiting. And Dr. Jovanovic says, well, there's reason to believe that capsaicin is potentially harmful in reducing the occurrence of this. And I have to admit to you that I would recommend that, I think. However, my colleague across the hall, Dr. Rother, who is equally well-schooled, equally well-credentialed, who I have the utmost respect for, and who in fact is the head of our department, doesn't share that belief at all. So if you had gone to see Dr. Rother, she in fact would not be recommending to you that you try capsaicin. Well, the reality is that when we look at the data to try and determine who's right, we find we don't have the available data. We don't have the data that enables us to answer that question. So what we are doing is mounting a study, what's called a randomized clinical trial, in which the treatment that you are going to receive will be determined by chance. And we can recommend that to you, or we can offer that to you, because we don't feel your care is going to be disadvantaged by your participating in this study. Okay, that notion that sort of that scenario reflects was put forward in a seminal article in the New England Journal of Medicine by a colleague here at McGill, an individual, Benjamin Friedman, who was also the clinical ethicist at the Jewish General Hospital, as well as a professor, a full professor here at McGill, who introduced the notion of what's called clinical equipoise. And that's uncertainty within the expert community, not necessarily on the part of the individual investigator about the preferred treatment. There is a current or imminent conflict in the clinical community as to the treatment of choice. So this represented a substantial advance, and in fact is at the core of the treatment of the ethics of clinical trials in the Tri-Council policy statement that I mentioned to you a few slides back. Okay. The decision about whether a particular trial is appropriate to even be offered to patients, let alone the decision of whether a patient chooses to participate in the trial, the question of whether that trial can even be proposed to a patient is decided by what's called the Research Ethics Board. And that's a body of researchers which is supplemented with community members and others with specific expertise in ethics, law, biostatistics, epidemiology, established by an institution to review the ethical acceptability of research involving humans conducted within the institution's jurisdiction or under its auspices, and assess whether the risks and the benefits of the proposed research are in reasonable balance. And this governs any research that's conducted in the institution that receives funds from any of the research councils, even if it's privately sponsored research by a pharmaceutical company, it still has to appear, it still has to uphold to the Tri-Council policy statement. So there's multiple layers of oversight, and if things work as they should, the Research Ethics Board provides a way of operationalizing the principles that have been proposed for making sure that the research presented to prospective participants satisfies the required ethical norms. Okay, we're going to change focus a little bit now with regard to other aspects of trials than the ethics of human experimentation. Let me just note that, or recall for us, that what we really want to try to do is make a decision about a proposed treatment. We will start with a patient who's in a particular condition, administer the treatment, follow the patient till a particular time period or the occurrence of an event, and see the status of the patient at that point. And we'll do so for patients who have received various competing treatments and try and make a decision about their relative merits. Well, one of the things to note is that there are indirect components of change or indirect components of the outcome that we observe. They are independent potentially of the actual treatment that was administered. One will just be the natural course of the disease. With the passage of time, there's going to be a change in the situation for the patient. In cases of disease, it's likely going to be a deterioration. In cases of prevention, it may be static or it may be an improvement, but time itself is going to contribute to changes. There will be another component of change that could result from other interventions that are administered during that period of time. And there's also a psychic component, the anticipation of benefit that comes from knowledge of participation in a study in which one expects that what's being done will hopefully produce a beneficial effect. What we'd like to do is to control for these indirect effects when we're making a decision about the effect of the treatment. So that means what we're going to want to do is introduce a control and make our comparison of the experimental treatment against the control treatment. So our assessment is going to be a relative assessment. So here we see the world of the turtle and the snail, where relative to the turtle, the snail is just a speed demon. On an absolute basis, we would think both of them are relatively tranquil in terms of the rate which they progress down their paths. But the issue is really how relatively one appears to the other. So when we don't have an available treatment, we don't have a standard of care to make our comparison with, we still would like to introduce some control for the effects we were describing, the indirect effects we were describing. And that's done through the use of a placebo to control for what's called the placebo effect, or we could call the comparator effect in terms of the broader context we were looking at in the previous slide. And a placebo effect is just the beneficial physiological or psychological changes associated with the use of inert medications, sham procedures, or in response to therapeutic encounters and symbols. And there's a lot of research that's done on placebos and placebo effects. They are an essential component of research. And one of the underlying questions for us is, when is it appropriate to use a placebo in a clinical trial and when is it not? From a regulatory point of view, regulators are prone to encourage placebo usage because it makes the analysis for them of the decision about licensure for a new agent relatively cleaner. And this gets us into a realm that's really more complex than we can deal with at this point. I'll just mention that there's been concern from an ethical point of view about overuse of placebos when in fact active comparators were available. So that's been an ongoing topic of debate, and there's a fairly large literature about that. If we use a placebo, and even in situations where we're using an active comparator, in order to maintain the benefits of that placebo usage, we need to ensure that, or attempt to ensure that individuals are not aware that they are receiving a placebo. That doesn't mean we're going to be deceptive in the informed consent. It just means that we're in the informed consent, we would tell individuals what the possible range of treatments that they might receive would be. And we would mention that it would be a placebo. But in the actual conduct of the trial, we would also inform the individuals, the patients, that they are not going to know what treatment they received. And we would try and construct a placebo that was similar in taste and appearance to the active treatment. And such a study would be described if it was only the patients who were unaware of what treatment was being received, as a single blind study. If, in addition, the treatment administrators, the caregivers and the outcome assessors were also unaware of the treatment that was that an individual patient had, that is an example of what would be a double-blind study. And again, it's an attempt to protect the study from bias that otherwise might occur if individuals were making assessments with the knowledge that a patient was receiving, say, the active treatment or a placebo. Let's take a look at different trial architectures, and we'll spend more time on the first, the parallel design, which is really the most common, probably on the order of 70% or more of clinical trials are done using a parallel design. The other designs are just listed here, and you can further check on them at your leisure, but we're not going to have the time to go into them right now. In a parallel design, we have a clinical trial that compares two or more groups of patients treated separately, but in this same study as part of the same study. And the inference and the comparison is made on the basis of a between patient comparison, because different patients receive different treatments. Hence the concern we have with assembling our groups in an unbiased manner. So we've basically or implicitly, I've been using parallel trials as the example as we've been going forward. But here's a sort of graphic representation of what happens, and the name parallel trial sort of reflects the fact that we proceed under parallel courses for the, let's use the example of two treatments being compared. So we assess eligibility. Recall, we want to make sure that a potential candidate is able to receive either of the treatments under consideration. We approach patients for consent. Those who are found to be eligible and who wish to participate are then randomized to receive capsaicin or to receive a capsaicin placebo. And then we follow the course of the patient's experience. We have a variety of factors that might come into play from the allocation down to the final analysis. Some patients may in fact not administer the treatment in the way that we hoped. Other patients may in fact choose not to continue in the study and withdraw. And we'll be left at the end of the day with the available patients at the end of follow-up for analysis in the capsaicin group, and similarly the patients in the capsaicin placebo group. While a trial is ongoing, we have a variety of concerns that need to be monitored, and typically those will be monitored by an independent committee, independent of the researchers who are carrying out the study. And that data safety and monitoring committee is charged with monitoring the ongoing safety of the study. They will be looking to see if there's any unexpected side effects that manifest themselves during the study, and also to see whether the study in fact has reached an answer earlier than would have been anticipated. When a study is designed, there's various assumptions that are made about the effect of the agent and, oh, sorry, I apologize. And whether, and the amount of information that would be required to give the desired assurances that we reach a correct conclusion. It could be that the effect of the treatment is even greater than anticipated, which means that it becomes manifest at an acceptable level prior to what we had anticipated would be required, in which case the study can be stopped. And that's a very fortuitous experience when it occurs. In addition, there are aspects of the conduct of the study that need to be monitored. It could be that the study is recruiting patients at a much slower rate than anticipated, and that procedures should be potentially altered, or other centers invited to participate in the study to increase the accrual. At the end of the study, we want to communicate the results to the scientific community, and the manner of doing so has sometimes been less than optimal. So a while back, and a while in this case means basically about in the past 20 years or so, a group of independent methodologists developed what was called the consort statement, the consolidated standards of reporting for trials, to help researchers improve reporting. It uses a checklist and a flow diagram to aid the understanding of a trial's conduct and to assess the validity of its results. Recall, as we looked at those little flow charts that we saw in a previous slide, we could have, if you like, various leakages from the study, individuals who actually withdrew from the study, some individuals who in essence withdrew from treatment but didn't tell us, they just didn't take the medication. And so these all present challenges to an assessment of, does this treatment work if in fact it's taken in the manner that's intended. So you could easily wind up with extensive arguments in the scientific literature between proponents and opponents of the particular treatment, and that, if it's based on a misunderstanding of what actually took place, generates a lot of heat, but not necessarily much light. So we try to make sure that the information that's communicated about a trial is clear, and that the decision that's ultimately reached leads to a correct process. Results from randomized trials should be available to the scientific community. It's typically easy if one has a positive result in a trial to get it published in a good journal, if it's a good trial, if it's a good question. If it's a negative trial, it's not necessarily the case that it will be that easy to be published. If it's a very, very large study for a very important problem, yes. But in other situations, that may not be the case. Nonetheless, it's important that the community be aware of the findings, if for no reason other than to avoid fruitless research continuing in an area which has already been shown not to be fruitful. So in a second, we're going to see some of the mechanisms that are used to ensure that that takes place. First, let's finish with this slide, which really relates to publication in scientific peer-reviewed journals. And peer review is a process by which experts review the report of a trial before it's published in a journal to ensure that the analysis and conclusions are sound. So it's a back and forth, and it can go several rounds between the authors of the publication and the reviewers for the journal, or the editorial board for the journal, to try and resolve any ambiguities or any frailties that are felt to exist in the reporting of the trial. If the results are particularly important, they may be featured in the news and discussed at scientific meetings and by patient advocacy groups before or after they're published in a scientific journal. Once a new approach has been proven safe and effective in a clinical trial, it may become a new standard of medical practice. I've listed here several, I've called them useful links. They're links to further information about trials, which all have very useful additional features. I've already mentioned to you the consort statement, and there's a variety of material available at the consort website about it, along with examples. We've talked about the Tri-Council policy statement. There's material available on the CDER's Can Support website on clinical trials. And the first item that's listed here, clinicaltrials.gov, is a wonderful website that's under the maintenance of the National Library of Medicine in the United States, which is a listing of clinical trials. Let's take a look. This is just a slight glimpse of some of the features of it. One is a registry of trials that are being carried out. Again, I don't have time to go into full details, but not too long ago, it was hard to obtain information, not only about results from some trials, but even whether a trial was being done. And there was a joint committee of Health Canada and the Canadian Institute of Health Research looking at appropriate placebo usage in Canada. And one of the questions that came up was whether certain trials were being done. And at that point, the Health Canada representative said, well, we can't reveal that. That's privileged information. Luckily, that view has changed dramatically, and it's appreciated now that information about trials should be available. And you can see that clinicaltrials.gov at this point has over 400,000 research studies. These are not all trials in all of the 50 states in the US and in 220 different countries. And you can search if you're interested in the website for particular diseases in particular locales. Once you put in the country, for example, if we put in Canada, we'll get the choice of locales. We can enter the country. And this next slide is a screenshot of what happens when we put in prostate cancer and Canada, Montreal, and you can see there's a listing of 199 studies, and there's further filters that can be employed to cut down that that list of studies to something more manageable. You don't have to be overwhelmed by the number. There's also, let me just point out, a section here, a tab about studies, and this contains useful further information to help you in your education about trials. And one of those is a link to a series of material that I'm going to ask Rola to now take over once I give her back the screen.
Hello. The Federal Office for Human Research Protections, or OHRP, created these videos to help you learn more about participating in research. Deciding if you want to volunteer in research can be difficult, and that the decision can have important consequences. To help you think it through, this video suggests questions you might want to ask the researchers and reminds you that your participation is voluntary. There are many different types of research, and the type of procedures done affect the risks involved, if any. The following questions might be helpful to consider when you meet with a researcher to learn about volunteering for a study. Perhaps the first questions you'll want to ask are, what is the study about, and what would you have to do as part of the research? The researchers should be able to explain in a clear and easy to understand way why they are doing the study, why you are being asked to participate, and what you should expect. You might also want to ask, how much time will it take? Will there be any risks or unpleasant side effects? Who will have access to your private information and how will it be protected? If it is a medical study, how will this differ from your usual care? Having a clear understanding of the study and any associated risks is critical to making an informed decision. Aside from understanding what the research involves, you should also find out whether participating will help you personally. For example, whether participating might help you with better monitoring of your disease. Researchers are required to tell you about the risks and potential benefits. Even if the main reason you want to volunteer is to help others, it's still reasonable to find out if taking part in the research may help you in some way. But remember, if the research is finding out whether an intervention works, don't assume that you will benefit from the intervention by participating. It is being studied precisely because the researchers don't know if it works. And if it's an RCT, the randomized assignment might mean that you end up in the group that doesn't receive the study medication or intervention. Another important question to ask the researcher is, what are the alternatives to participating in the study? Would the same treatments or procedures be available to you if you choose not to participate? How would joining the study change your treatment options? Having the answers to these questions will help you weigh all of your options in light of the study's risks and potential benefits.